Hi, Dave. Welcome to the podcast. Why don't you tell us about your role at Eurofins and what you do for the company?
Hi, Karen. Yes, so I'm the Director for Scientific Strategy at the Eurofins Food Chemistry Testing Laboratory in Madison, Wisconsin. So predominantly in charge of overseeing a lot of the method development in the macronutrients business unit, but I also contribute as well to some of the other business units such as inorganics and specialty testing and vitamins.
Okay, so this episode is about human milk oligosaccharides, which you're a subject matter expert in. So why don't we start at the very beginning? What are HMOs and why are they so important to the infant formula diet?
Yeah. So HMOs are non-digestible carbohydrates, typically made up of 3 to 30 units of monosaccharides. Those 5 different sugars that can be found are galactose, glucose, N-acetylglucosamine, N-acetyl-neuramic acid, and fucose. And there are various structures. Typically, there are 3 general categories. Fucosylated is one, obviously containing a fucose moiety. Sialic is another one containing a sialic acid. And then you have your neutrals, and that is typically just containing galactose, glucose, and acetylglucosamine. There have been over 200 different structures to date that have been found. And that number continues to increase based on the research. The benefits of HMOs, predominantly serves as a prebiotic. So for the intestinal microflora, right? So prebiotic, and then how does it function in terms of the benefit in the human gut? It serves kind of as inhibitory for viruses, pathogens on the intestinal epithelial cell wall. So it can serve as a blocker. It can serve as a decoy, right? The sialic acid forms can also serve for brain development for that sialic acid moiety. A fun little tidbit on the sialic acid forms: in certain mammals, you will have different versions of that sialic acid. In humans and bovine, you're going to have N-acetyl-neuramic acid, whereas in sheep, if you fortify with sheep or goat, you can find N-glycol-neuramic acid. So these are different compounds, one of the many that are there, but in general, that's probably a good background. I think another interesting thing that most people don't think about with HMOs is they're actually the 3rd most abundant solid in human milk, which after lipids and lactose, there's actually more HMOs than protein.
Yeah. Okay, so can you tell us why infant formula manufacturers want to fortify their product with HMOs and why we see different variation of the HMO products in formulas?
Yeah. So the prevalence of certain HMOs in human milk differs, right? The top 10 HMOs cover 70% of the total profile. And if we look at 2'-fucosylactose, which was the first HMO that came out, right? That covers 30% of the whole profile. And so when you look at that, there's obviously a reason why some are more prevalent than others.
Sure.
And that comes, goes towards the benefit of and the function of these HMOs. And so for a manufacturer, I'm going to try to replicate the most common, right?
Yeah.
It's as close as I can get to the mother's milk as possible, right? And with production of these HMOs, it's not. It can be costly, right? And that's kind of a limiting factor. And what you'll see in different infant formulas is substitutions for these prebiotics. And so you'll see galacto-oligosaccharides, you'll see fructo-oligosaccharides, or polydextrose, right? And these are more cost-effective ways that they can provide a prebiotic in the formula.
Yeah.
But as production processes improve for HMOs, it becomes cheaper to be able to mass produce these HMOs and start adding them in. But we're priority wise, we're going to start from the most prevalent and work our way down.
Yeah. So Eurofins has a method, obviously, for, or maybe not obviously, but Eurofins has a method for testing HMOs in infant formula. What is the scope of our method as far as which HMOs we can quantify, and then and what matrices we can test it in? And then also, are there any ingredients that can interfere that may be in the formula that can cause an interference in the
Sure.
method?
Yeah, so I'll start out with just maybe a background on our method
Yeah.
itself. The method is based on derivatization of the HMO with 2-aminobenzamide. So it adds a fluorophore to the compound. And what that does, it provides a lot of specificity and sensitivity to the analysis when we start working with fluorescence. Once we derivatize them, then we separate those out by HILIC, hydrophilic interaction chromatography, and then detection by fluorescence. The method that we have, it covers 6 different HMOs, 5 in infant formula, one of them difucosylactose. We do include in certain supplements that we can test for that in the current method. The 5 main ones are 2' fucosolactose, 3 fucosolactose, 3' sialactose, 6' sialactose, lacto-N-tetraose, and lacto-N-neotetraose. The other ingredients, so infant formula being very complex, right? So they add a lot of other different carbohydrates into the formula. And I guess the benefit of this type of derivatization is it really only targets reducing terminal end sugars. So that kind of rule out fructooligosaccharides. Those are not an issue. Galactooligosaccharides, yes, it will label those, but lo and behold, galactooligosaccharides actually contain, as one of their main components, some of the galactosylactose, the 3', 4', and 6'. And those are recognized as HMOs. Only when we have analyzing at very low concentrations will we have potential interference maybe on some of the late eluders in the chromatography with galactooligosaccharides. Same thing with like hydrolyzed casein. You might see some interference down towards very low levels. But in terms of products, we took our method through the stakeholder panel on infant formula adult nutrition. So there was a variety of different products that were tested through that.
Sure.
And it came through with flying colors on the majority of these products, and demonstrated, obviously, getting to first action status, it can meet the requirements to quantitate in all those products.
Yep.
So again, all the different variations that you can think of in infant formulas, it can test in.
Yeah. So you touched on it a little bit there. The Eurofins method has made first action in 2022. Congratulations. Where are we at with getting the method to final action?
Yep. So the expert review panel decided, and this was after our method had received first action. There was another method that came in almost identical to ours, but it does use ultra high performance liquid chromatography or ultra high pressure. And other than that, the chemistry and the format procedures are generally the same. And so they decided that we either pick one method to move forward to a multi-lab validation. And so Eurofins and the other authors, being Nestle, we decided to collaborate and take the best of both methods and make a merged method. And we have accomplished that. And we are currently undergoing internal validation to actually update our method too as well, for using this merged method. The benefits of the merge method is the increased resolution in the chromatography, and also the addition now that we can analyze difucosylactose, and potentially some other HMOs that manufacturers are going to be coming out with. So it does open us up and I think provide the industry a very good method And it's, I guess, something that most laboratories can set up as well and have available to use.
That's fantastic.
Yep.
So where are we with timeline on getting the merged method offered at
Yeah, so
Eurofins?
we do plan to present this method to the expert review panel in the spring
Hmm.
during the Analytical Methods Week. And then upon a granted approval, right, we would move into a multi-lab at that point.
Yeah.
And so we'll be looking for labs to participate in that. Um, but for our routine method, we will be offering this in early spring of 2025.
Fantastic. I can't
Yep.
wait. All right. So, what do you think's next on the horizon for HMOs in regards to infant formula specifically?
Yeah, so I talked a little bit about some of the substitutions for prebiotics earlier.
Hmm.
I mean, I think that's going to continue for a little while yet as the processes for manufacturing these HMOs and at large scale improves. Once that happens, I think you're going to probably see a transition from the fructooligosaccharides, the polydextroses, the galactoligosaccharides to HMOs.
Yeah.
And those particular HMOs that they're interested in, again, I think they're going to be looking in the literature and the clinical studies to understand which ones, again, twofold, the most prevalent, but also providing some sort of health benefit, right,
Yeah.
for the humans.
Absolutely. Okay, so with the new HMOs that potentially could be coming, will our method, the merged method, will it be able to quantify those HMOs, or will we need to do further method development work?
Yeah. So the advantage of our separation is we can already see where a lot of these other HMOs elute.
Yep.
And so it is a minor modification to the elution, but we already kind of have a step ahead on where things are going to be and what's the possibility. With the hillock separation and the column technology that we have, there's a good chance that, a lot of the newer ones that are going to come out, we should be able to analyze and quantitate.
That's fantastic. Well, I think that's been super informative, Dave. I think HMOs are definitely going to be one of the top topics in the next few years for infant formula. I think it's going to replace the prebiotics that we discussed, GOS, FOS, polydextrose, etc. And I think it's just the start, really. So it's very exciting for carbohydrate chemistry. And I appreciate you coming onto the podcast.
Yeah. Thank you,
Thank you.
Karen.
Thank
Thank you
you.
for having me.
Absolutely.